All right, membranes and membrane structure function. In this video, we're going to be looking at a few aspects of structure, and the slide here points those major concepts out for us. We have two different models for membrane structure. The first is called the sandwich model, and you're going to want to be able to describe how the phospholipids, those lipids of the protein, and the proteins of the membrane are oriented in relation to one another for both of these models, both the sandwich model and the fluid mosaic model. And then also, what is the support? What is the scientific evidence that was put forth for the model? And then one more thing I don't have listed on this slide is whether or not the model is currently accepted. We don't say right or wrong because we can never ever know all of the information that could be out there, and maybe in the future our current understanding will change and there'll be new evidence and we'll have a completely different model of the membrane. So we always want to keep that in mind as we're looking at membrane structures and really any concept in science in general. You always want to accept, but you don't say it's right. It's just kind of almost like a bad word. All right, so let's move on to the first model, Davison and Dinelli's sandwich model. And as you can see here, the orientation of the phospholipids in relation to the protein shown here at the bottom. We have our phospholipid bilayer, as we talked about in previous videos. And then these different colored structures, these circles, represent the proteins. So with the sandwich model, it was the phospholipid bilayer in the center, and then we had a layer of protein on the outside and another layer of protein on the inside of the membrane. And the evidence for this model was actually electron microscopy in the 1950s became available, and they took an electron micrograph of the cell membrane, and they saw these two dark lines, and they said, oh, well, this dark line here, that must be these outer membrane proteins, and this other dark line here, oh, that must be the inner membrane proteins, and then that light area in the center, that must be the phospholipid bilayer. So that was their reasoning or their evidence. Now this model is not currently accepted. The model that is currently accepted is the fluid mosaic model, where we have our phospholipid bilayer, just as we did in the other model, but this time we have the proteins embedded in the membrane, which was not the case in the other model, and they do interact with the phospholipids. Okay. Now the evidence for that model was the freeze fracture studies, and what they would do is freeze the cell, and then they would strike it with a blunt object, or here we actually have a knife. It doesn't have to be a sharp object. But the cell membrane would break down the point of least resistance, which would really be between those phospholipids, those fatty acid tails of the phospholipid bilayer. And looking at the extracellular layer, we saw these proteins popping through, and then looking at that cytoplasmic layer, again, the proteins coming through and giving evidence for this idea of proteins actually embedded in the membrane. All right, so let's move on and talk more about this membrane structure. So what makes our membrane fluid? Well, there's a few things. And the movement of the actual phospholipids in the membrane is one of them. So let's take a quick look at an animation that we have on Isidore. And if you go into the chapter 7, or the membrane chapter folder, we'll click on membrane fluidity here, and it'll bring up a lovely animation, which I personally love. So here I'll orient you to the diagram, or the animation. Our phospholipid bilayer, so here's our phospholipid, here's the other layer, the phospholipid bilayer, the membrane. Here we have a membrane protein that spans the entire membrane from outside to inside. Here we have another protein. It's also embedded, but it doesn't span the entire membrane. So there are two types, the more peripheral and then the integral or transmembrane proteins. So let's take a look at the fluidity. All right, lovely. So as you can see, the phospholipids and the proteins. This is worth a second look. All right, the phospholipids and the proteins move in relation to one another. So it is a dynamic structure. It's not static. It's not just standing still. And as you have probably seen in the past, you draw a cell and you just draw this solid line, and it's really boring and doesn't look like anything's going on. But the membrane, my friends, it is dynamic. Okay, so let us go back to the PowerPoint itself, talk more about structure. Another aspect of membrane structure that is important is the idea of the saturation or the unsaturation, as it might be, of those fatty acid tails of the phospholipids. And then also cholesterol plays a major role in your membrane. Cholesterol gets a really bad rap, but cholesterol is absolutely vital for life, absolutely vital. Now, when it gets out of control or you have too much circulating in your blood, it can get into trouble. But again, cholesterol is absolutely essential and vital for life, and it does play a major role in membrane structure. So let's take a look. So this is just a diagram showing saturated fatty acid tails, and in the chapter on lipids, you looked at the saturated versus the unsaturated. So saturated means there's hydrogens all along those carbons. Everywhere there's a place to bond with carbon in the fatty acid tail, hydrogen is there, making that tail very straight. And as a result, the tails are straight, they are very close together, and they form more of a solid structure or less fluidity. And over here on the right, we have these points of unsaturation, double bonds, and as you might know or should know by now, whenever you have a double bond, it creates kind of a kink in the chain. And as a result, you have these spaces that form. And when you have space, my friends, oh, we have fluidity. So more unsaturated fats, you have more fluidity. And as we looked in the macromolecule chapter, any kind of oil, plant oil in particular, is going to be liquid at room temperature. So you have your olive oils or your corn oils, and those are liquid because they are highly unsaturated fats. Whereas animal fats, like butter, that is a saturated fat, so it is solid at room temperature. Okay. All right. So here's a question for us, and there's something similar in your assignment and in your study guides this week. But looking at this question, which of the following factors would tend to decrease membrane fluidity, so make it less fluid? So more unsaturated fatty acid, phospholipid tails, or more saturated? So it's one or the other, it can't be both. So remember we said that unsaturated leads to more space, more fluidity. So that would actually increase fluidity. So we can't say that's the correct or a possible option, so we'll cut out A. Now B, more saturated, remember those are nice and straight, they are really close together, so that would decrease membrane fluidity. And then temperature. The higher the temperature, the faster molecules, atoms move around. So high temperatures would tend to increase fluidity of these fats, whereas low temperatures would of course decrease. So I guess B and D would be our answers. Okay, so I'm going to end the video here, and we're going to pick up in part 2 looking at membrane function.